Hi, everyone, and welcome to our September meeting. This was our first all video meeting in quite a while. I know we've got a good program for everyone. I hope everyone had a good summer, first of all, and survived the heat wave, and hopefully got some rain in the meantime. We have a couple of announcements, but before we get there, I wanted to ask if we have any new members. And if so, please introduce yourselves on the chat with your full name or name that you want to go by, general location, and plants you are interested in. The next announcement is that Laura has set up a really neat field trip opportunity. This is on October 23rd to Ricardo's Nursery in Long Beach. I haven't yet met Ricardo, but I've interacted with him by text, and I've heard actually quite a lot about him. I know he's quite interested in some rare mango cultivars, and it sounds like he's got quite the operation going. And it sounds like an interesting guy overall, and I was hoping to meet him. I think this is a really neat opportunity to see what he has. The nursery itself has won a handful of awards, and he's extended the invitation. So Laura has set this up. It's October 23rd. The details will be on our website. And over the next week, look for signups and more information there. And then we otherwise have two more announcements. Martha will make one, and followed by John McNeil.
Hi, everybody. I wanted to let you all know that the propagation committee is going to meet on September 24th. We're going to be meeting at Norm's. It'll be one of our usual meetings, and we're going to be up-potting a lot of little seedlings that have outgrown their pots. So everybody is invited. If you have any questions, give me a call. I have sent the message out to all the propagation committee meeting members. And if anybody would like to join us that is not on that list, you can get in touch with me and I will be happy to give you the information. Again, that'll be September 24th. We'll meet at 9:30 in the morning. Probably get underway closer to 10 o'clock. And we will be having our usual potluck luncheon, which everybody enjoys. So I hope to see you there. Please give me a call or email if you have any questions, and I turn it back over to Khaled.
All right. John McNeil?
Yep. Okay. Thanks, Khaled. Welcome, everybody. I have, during my chairmanship, I was granted permission to create a chairman's award to be presented by the outgoing chairman to an individual who has made a significant contribution to the chapter and has been approved by the executive committee. Tonight, I am going to make that presentation. So please stay tuned to the end of the presentation to find out who I'm going to make the award to. Now, just for your interest, this is the award. I'm not sure whether you can see it coming up. It is a plaque. This plaque was created by Jay Kreuzinger, who is a member of the Orange County Organic Gardeners Club. And he graciously took a design and created this beautiful piece of art. Okay, so I'm going to make this presentation this evening at the conclusion of our our speaker's presentation, and stay tuned. Okay, thank you, Jeff.
Well, thank you, John. And now, without further ado, I'll turn this over to Jeff to proceed and introduce our speaker.
Okay, great. Tonight we have Herb Machleder coming to speak to us about plant hormones. I know Herb from the Master Gardener program in LA County, where I took the courses in 2017, and Herb was hands down my favorite instructor. As my critique afterwards that we had to do on all the instructors would have shown at the time. Herb is a retired, he's professor, emeritus professor in the Geffen School of Medicine at UCLA. He became a master gardener in LA in 2006, and in 2007 he did this deep dive into orchard management by taking a course at UC Santa Cruz at the he became a resident farm apprentice at the Center for Agroecology and Sustainable Food Systems. So within the Master Gardener program, Herb created the orchard team. He's been very involved in creating, installing school, a community garden, and other types of community settings, orchards into these settings, and figuring out what fruit trees do best in those settings, so that the crops are coming in while the students are there, and so the maintenance isn't complaining about all the fruit dropping on the ground during the summer. In any case, that group's done quite a bit. So with no further delays, here's Herb. So you're muted, Herb. There we go.
I do my best work when I'm muted. Anyway, I have a little 4-minute video introduction, which was done in the morning. So it says, it starts with good morning, but I am oriented. I'm going to share my screen. Before I took over at the Master Gardener program, I had toured a lot of the Central Valley gardens with the farm advisors, maybe for 3 years. And then I worked in orchards in the Central Coast. And it appeared to me that that these big orchards, even 10,000 acres, were very easily managed, really, compared to the backyard orchard or the home orchard or the community orchard. And it dawned on me that the trees were all clones, so that if you brought a pruning squad in and demonstrated how to prune one tree, they could go through the entire orchard and do it. But a home orchard, the orchardist is going from a pear to a plum to a peach to a persimmon, an apple, and also an avocado and a citrus. And each one of them has different requirements, have slightly different fertilization needs, water needs, harvest times, pruning requirements. And it's very confusing. And for the 10 years that I've been leading this Master Gardener orchard team and going around orchards primarily in Los Angeles County, although I did graft the orchard at the biggest little farm in Ventura, stepping out of my Los Angeles role. But anyway, I was really surprised that the crops were not very good. They were very erratic. The fruit wasn't of the quality it really should be. And I recognized that the differences is what was confusing to people. But the hormones are the same in all the trees. And if one understands the role of the hormones and how they affect the tree's management, it becomes a lot easier to manage a diverse orchard. The other thing is there's a lot of homologies between all kinds of biologic systems facing the same environmental stresses. And people don't recognize that. They think of them as discrete entities, but nature is very fastidious. Most organisms face the same environmental problems, whether they be day and night, seasons, climate, microorganisms. For example, Pseudomonas is one of the most common organisms in the tree cankers, but it also is a very common organism in human urinary tract infections. With hormones, we have something like melatonin that helps us separate day and night, and the hort trees have 2 hormones that help them analyze the photoperiod, the same as we do. We require a rest period, they require the period of dormancy. So I'm going to start off with a little, I just want to mention that because I do draw these analogies from time to time between biological systems and orchards so that you get an idea that these things work very similarly in the environment. Now, the first thing is a little introduction. And it was done in the morning at the South Bay chapter, so you'll have to excuse that, but I didn't want to do it over. Good morning. I'm Herb Machleder, an instructor with the University of California, Los Angeles County Master Gardener Orchard Team. And the subject we've picked for this morning's meeting is how a greater appreciation for the actions of the plant hormones can dramatically improve the management of our urban and backyard or home orchards. We're at the monthly meeting of the South Bay chapter of the California Rare Fruit Growers in the waning months of the year of the COVID virus. If any of you have had the opportunity to visit a high-density modern orchard and a contemporary packing facility in the Central Valley, you certainly would appreciate the advance of precision orchard management. In a high-density apple orchard, the feathered trees with many fruiting spurs are prepared in the nursery by inhibiting the hormone auxin using a cytochrome. All the trees in a block uniformly come out of dormancy by reversing the hormone abscissin. Blossoms and fruit load are controlled by precise hormonal thinning. Fruit is kept from dropping prematurely by blocking ethylene, using retain, and uniformly released for harvest. They're then optimally ripened with full color by applying the hormone ethylene. Avocado orchards in Santa Barbara, Ventura, Riverside County are helicopter sprayed with the naturally produced hormone gibberellin, which increases the yield of market-sized fruit, 40, 48, and 60, by more than 50%. Crop of 40,000 pounds per acre, that's 110 trees, can be inspected after this application of gibberellin, and this is approved and registered for certified organic production. What is the driver of this need for precision? Well, Washington has 175,000 acres of apples, producing 2.3 million tons of apples per season. California has 1,530,000 acres of almonds, more than 400,000 acres of walnuts, and even more than 25,000 acres of apples. Management of the plant hormones and the PGRs is fundamental to success in growing, harvesting, and marketing tree fruit. Harvesting labor and equipment, trucks, and a slot at the packing house all have to be prepared in advance. If the average high-density apple orchard produces, depending on the variety, between 30 and 50 tons of apples per acre. So even a small farmer with 10 acres of apples can't bring 500 tons of apples to market without a precise management and pre-planning. Well, this type of commercial management is not what we're interested in. It's not why we have our home orchards, and we don't teach this in the Master Gardener program. But the goal and the purpose of the Master Gardener programs, supported by the agricultural colleges and universities, is that we bring all of this horticultural research to the urban grower so that they too can benefit. After all, it's the urban growers who are supporting all this research in the agricultural colleges. I think you'll be impressed with what you learned this morning and what you can do with this information. And there won't be a cent spent for chemicals. And if you want to purchase a tool, it'll cost no more than a coated cardboard cup of macchiato grande, whatever that is. This is not going to be a chemistry lecture. The discussion is going to be down to earth, and all the examples we use will be those that come from small urban orchards, just like yours. Okay. The first thing is the classification of the plant hormones, just so you understand the names, because we're going to talk about each one and what they do and how we control them with our physical production in our orchards. Auxin, a very important hormone that's produced in the meristem, that's the growing group of cells, particularly in the apical buds and young leaves of trees and plants. Cytokine is another very important enzyme that's produced primarily in the roots. Gibberellins are very important in the embryos of developing fruit and have a very important role in keeping track of the photoperiod and also are very important in terms of alternate bearing. Abscisic acid is produced throughout the tree and is a very important stress hormone and seasonal hormone, and ethylene, the most important gaseous hormone for the transfer of mature fruit to ripe fruit, which is, of course, is not the same thing. So they're also classified as growth promoters, the cytokines, the gibberellins, and auxin at low concentrations. And the ones that retard growth are abscisic acid and ethylene. Well, that's enough about the names, and you'll recognize them more as we talk about them. Abscisic acid, let's first look at abscisic acid. Since that's the dominant hormone of autumn, and now is being developed in all of our trees, particularly those that come from the northern part of the hemisphere, the deciduous trees. What stimulates abscissin and sets it in motion? Well, this hormone keeps track of the photoperiod. And when the autumnal equinox comes, the day and night are the same hours, and then the day hours become shorter and the night hours become longer. The tree, by this mechanism, using this hormone, recognizes autumn and also recognizes the vernal equinox, which is spring. So this is the hormone that starts and maintains dormancy. So it's stimulated by the light, the photoperiod, and the onset of what we call autumn. What does it do? It initiates the onset of dormancy. It sets a terminal bud, which is one of the first indications of dormancy. The small group of trees at the end of each shoot become a bud, which is a predetermined bud in that it has all of the sites that will grow the whole year, and it sets the limit of growth for that season, for that next summer season. It also causes abscission of the leaves, where it gets its name. It's same root as scissors. It was believed that they actually cut off the leaves. But the way it does this is it actually works the same as our stress hormone adrenaline. It causes a contraction of the vessels. The phloem coming down from the tree, analogous to our venous system, and the xylem going up from the ground, bringing nutrients to the leaves, which is analogous to our arterial system. And it does this by causing a proliferation of the cells in the petiole until it blocks the flow, particularly of the xylem. It also sets in motion, prior to C, it sets in motion the translocation of metabolites so that the leaf color gradually changes. The chlorophyll is broken down first, and you begin to see the other photoactive hormones, the anthocyanin, the red, and the xanthophylls, which are yellow, so that the colors are all there. They react to different wavelengths of light, so that the tree can take advantage of light from different wavelengths in going through photosynthesis. The abscissin then maintains dormancy until the onset of spring, and the amount of abscissin in each tree is dependent on its genetics, which are developed at the site where that tree originally developed. And it's the amount of abscissin that's in the tree that it accounts for what is called our dormant period. And we'll go into that in more detail. Sometimes you see that leaves stay on the trees. The apples never seem to go dormant. The question is, can we manipulate abscissin to assist our trees in achieving dormancy? Without a dormant period, the blossoming out in the spring is very erratic and not very effective. And the crops then vary very dramatically from year to year. Lots of questions surround the actions of abscissin. Why do some trees leaf out in the winter and then blossoms die? Why do some trees set blossoms in late summer and the fruit never ripens? What exactly are chill hours and how do they affect our orchards? These are the considerations one needs to address when discussing abscissin. So first, just to review, what are chill hours all about? Well, the number of chill hours and the water requirement for an index crop are downloaded daily from 140 active automated CIMMYS, California Irrigation Management Information Systems, weather stations located throughout California. The cumulative chill hours are entered on the web daily between November 1st and February 29th. 80 of these stations have historic number of chill hours, so you can see what for your area is the average number of chill hours that can be recorded at that site. This is a page from the Fruit and Nut Research and Information Center at the University of California, Davis. And here is the interesting thing. Between 32 degrees Fahrenheit and 45 degrees Fahrenheit, the trees accumulate or aggregate chill hours from November 1st through February 28th. When it's colder than that, the enzyme that degrades the abscissin does not function. And if the temperature is above that, the enzyme that degrades abscissin doesn't function. So it basically, that tree will have a residual of abscissin come spring, and it doesn't blossom out readily until it acquires heat units. And this accounts for the late blooming of many trees that have an asymmetric relationship to the chill hours that they have in their genetics and are found at the site. Interestingly enough, below 32 degrees Fahrenheit, the chill hours are not accumulated. So some of the very, very northern growing sites have actually low chill apples that we can grow here, because sometimes during the month of February in upstate New York and northern Michigan, it never reaches 32 degrees Fahrenheit, and it may be below zero most of that time. So that the chill hours are kind of critical in deciding what trees will do best at any particular site. But it's between those two temperature gradients. Most hormones and enzymes operate ideally and almost exclusively in very narrow ranges of pH and temperature. Los Angeles, these are the residual stations. Los Angeles in the 20s and 30s was one of the biggest producers of orchard fruit in the United States, and has all of these CIMMYS stations that are still functioning. Here's an example of the difference. In Los Angeles, for example, where I've been working, in LA County alone, we have every chill hour profile in the United States, from 24 chill hours per year in Santa Monica to 1,357 chill hours accumulated yearly in Palmdale. Though people in different cities cannot really discuss with one another, it's different in Orange County. You have a much narrower range. But in many of the counties in California, up and down the coast and in the Central Valley, from one site to another, the chill hours are so different that the cropping, the harvest times, and many of these characteristics of fruit trees are very different, and account for some of the disparities that people don't seem to understand very well. Here is a typical graph of the chill hours. This is for Long Beach, which is on the northern part of your area, and it shows you each one of these different colors is a different year, 2014 to 2019. And you can see the chill hours do vary. It's not a huge variation, but it's between almost 200 and 400 chill hours accumulated. And this largely accounts for the difference in the cropping and the harvesting in different years. The fertilization and water application in most orchards is generally the same and very easily managed, actually. It's the chill hours that accumulate differently, and this is entirely dependent on the hormone abscissin. Here's your county. This is Irvine and Coto de Casa. You can see Irvine has a narrower range. Coto de Casa has a higher range. Also, it has areas that accumulate a lot more chill hours. Under 300 chill hours is considered low chill. Many nurseries cut corners by describing their fruit trees as low chill, or they don't indicate what the chill requirement is. But it is critical if you want to grow fruit and have a productive orchard. This shows you how the chill hours accumulate. It's almost linearly, although there is variation from week to week and month to month. But nevertheless, the end result is a fairly narrow range. What can we expect if we get the chill hours right? Well, once the chill hours are met, the blossoms begin to open. And there you have it. That's the message. When the chill hours are for that tree are met, it means that there has been enough time and temperature to allow the enzyme to degrade all of the abscissin, which is what's maintaining that tree in dormancy. What if I get the chill hours wrong? Well, everything grows here. You'll probably have a lovely landscape tree, but the cropping will be very erratic, and sometimes you'll get no crops at all. And as I travel around to different orchards over the past 10 years, there are people who have lovely trees, but they never have any crop. And I try to figure out whether it's nutrition. The leaf analysis shows that they've been taking good care of the tree. If I can track the variety back or call the nursery where it originated and look up the chill hours, that generally will tell the tale. This is what you're going to expect if your chill hours match your orchard site. This is a 10,000 acre orchard. The trees all blossom out. This could be a peach or an almond. They blossom out within hours of each other because they are clones and their hormonal structure is identical. And the temperature variant in this 10,000 acre orchard is minimal. The difference in our urban orchards is that trees will have different chill hours accumulated depending on where the branch is. And very often you'll see branches that are out in the sunlight compared to branches that are in the shade accumulate chill hours at a different rate. And it accounts for the stuttering blooming of many of our trees. Here's a recent picture. An orchard acquires more chill hours than needed by the fruit variety. So what happens? Well, the tree blooms on an occasional warm day in midwinter. You have a December day when it's 80 degrees, the tree begins to blossom out, but there's not enough abscisic acid remaining to maintain dormancy. It gets colder in January, the blossoms fall off, there's no fertilizers or insects around, and what happens is you get a very poor crop. These apples that are forming now in some of the trees in my orchard are not going to ripen. There's just not enough time. If an orchard has inadequate chill hours for that needed by the fruit variety, the tree blooms later in summer, stimulated by heat units, doesn't have enough time to ripen the fruit. So when there's a dissimilarity or a disparity between the chill hours required by the tree and that acquired by the site, you're going to have an erratic harvest at best. Here's an example of, I was asked to give a lecture for Earth Day at Santa Monica City College, late April, and I decided to pick up a tree at a local nursery, a very prominent nursery, just to use it as a prop. It was in a pot already, so I know it didn't sell because this nursery sells bare root trees. When they pot them up, it means nobody bought it. I thought I could get a discount, but I couldn't. It's a taste test winner. That's a real bugaboo. That's very dangerous for the home grower, because this is a taste test winner acquiring chill hours in the northern part of the Central Valley. Well, I took it up to the desk and I said, "Can I get a discount on this?" And the salesperson said, No, this is an excellent tree. It just needs a little pruning." And I looked at it and I said, "Well, this requires 700 to 800 chill hours. The next time this thing is going to bloom in Santa Monica is during the next ice age. And it's pollinated by Santa Rosa or late Santa Rosa or another pluot. So unless the person has another tree in the area, it has a virgin birth maybe, but a parthenocardic birth, but it's not going to be a very productive tree." And this happens in a well-known nursery because they purchase trees that may do well in one part of the county, but not the other. Now, here's a good idea of how we can use the information about abscissin, because it is a stress hormone. And just note, this is a very important graph that's published by Agriculture and Natural Resources in the University of California, and it's the tree water use gallons of tree per day in California coastal and inland trees. And this is done with a lysimeter, where trees are grown in a huge shipping container in the ground and on a scale, so that they can measure the actual water loss every single day for all different kinds of trees and plants. And that's how this type of information is generated. It's very carefully done. But it gives you the water for June, July, and August, and April, May, and September. So there's no water for 6 months during the year, from October to April. And actually, it's the abscissin that enables you to withhold water and induce stress in the tree. And one way we can get our trees to go dormant is to withhold water after September. Is if there's enough groundwater for the roots, that's all it needs. It's not transpiring any water at all, because the abscissin closes the stomata. Those are the little openings on the undersurface of the leaf that allow it to take in atmospheric air and join carbon dioxide with water from the roots to form the carbohydrate. Once the stomata are closed, the tree loses no water, the leaves dry up and fall off, and it doesn't produce any carbohydrate. It's dormant. And this happens in even live oak trees when they're stressed. So it happens in all trees. It's the stress hormone. There's, as I mentioned before, a proliferation of the vascular cells in the abscission zone, which is in the petiole. So again, when we see our apple trees, if you're growing apples down in Orange County, you withhold water as long as there's some groundwater there and there's an occasional rain that provides some water for the roots to stay alive. That'll force the tree into dormancy, and that will give you a much better harvest when the tree leaves out in the spring. Trees are delayed in setting blossoms. Contracts have been let for labor, packing house, supermarket. What does a commercial orchard do to get things going? Well, they can block the residual abscisic acid using a PGR, a growth hormone called Dormex. This is hydrogen cyanamide. It blocks the effects of abscissin and its analogs, provides for earlier bud break and a more consistent harvest. In some crops, it can make up for a season with inadequate chill hours and stimulate earlier bud break. It's used very frequently in the southern part of the Central Valley, where they're growing cherries without enough chill hours to ensure a good harvest. They're manipulating the harvest date by controlling the hormones. So the enzyme, which is degrading abscissin, works between 32 and 45 degrees. When all the abscissin is gone, the buds begin to open. So the message there is try to match your site with the chill requirement of the trees, and you're off to a good start. Once you have a tree that doesn't match the chill hours, it's a losing battle. There's just no way of correcting it. Well, let's talk about auxin. This is one we can control very well. It's the queen of the plant hormones and of critical importance to the small orchard farmer. And I'll show you how we use this. It promotes and maintains apical dominance. Well, most of the northern trees grow straight up, suppressing lateral growth because they grow up in a forest, southern Russia, northern China for apples, and they need to get up into the sunlight or they won't survive. And that's what the tree tries to do the first 3 or 4 years, is to get up, to get light. So that's the apical dominance. It is controlled by auxin. So the auxin maintains what we call paradormancy in the axillary and axial buds. That's all the buds along the axial portion of the tree's trunk or its shoots. And these buds are in paradormancy because they're not in dormancy because of the season. They're being suppressed by auxin, which is gravitationally acted on. It acts in conjunction with light, with pressure, and with gravity. So it suppresses lateral branch induction. That's why it has to be suppressed if you're going to have nice feathered trees for a high density orchard. It directs vegetative shoot growth towards the sunlight. It turns leaves and flowers towards the source of light. It also affects our pruning cuts, heading and thinning cuts, if you understand how it's working. And it impacts summer and winter pruning very definitely. And it affects bud grafting very significantly. How can we control auxin organically and make it work for us? Can our biological manipulation of auxin affect our orchard management? Well, that's the story. We're all told that when you get a bare root tree, you plant it with the graft above the ground, then you prune it to a knee-high whip. Well, most people don't do that from the orchards I see. They bought the whole tree, they spent $60 or $120 for it. Why are we going to cut it down to a twig? Well, there's a very important reason. You have to know your buds. The buds at the tip are proleptic buds. That means all of the internodes are predetermined. Those shoots that come with that tree, which is grown as a liner, will grow to a certain length and then stop. Even though it's mid-July and you have August and September left to grow, they'll stop. Sileptic buds that grow along the trunk and also grow out from buds from laterals keep growing. As long as there's enough water and enough warmth, they keep growing. In a peach tree, they'll grow easily 3-4 feet in a season. Epicormic buds are buds that are beneath the epidermis or the bark of the plant and tree. If you see the pruning teams go through your neighborhood and hat rack the trees, within 2 weeks, there's all these sprouts coming out from the top, causes some nice shade below, but it's not a very organically looking tree. Epicormic buds are the tree's backup. If a catastrophic event happens and a limb breaks or it loses a part of its trunk, the epicormic buds are there waiting. They blossom out, shoot out, and they grow all summer and fall. So basically, if you control that, you get the entire architecture of your tree in the first season. Basically, if you cut this off, these epicormic nodes begin to blossom out and shoot out. So you can pick the ones you want. If you trim off the apical bud, which is producing the auxin, you'll begin to get the laterals. And basically, you can have the entire tree. It'll keep growing as long as there's warm weather. So the bare root trees, they arrive in January and February. They need to be pruned properly. They're grown like this because they're liners. They're grown like a field crop in a long line. And then they're harvested by a tractor with huge wheels and a big U-shaped knife that goes down the entire row, cuts all the trees off. They fall over, the roots are washed, and they're packaged up and brought to the retail nursery. Why are they sold like this if we have to cut them down? Well, depending on different types of orchards, you can grow them as a lateral or vertical Y. You can pick and choose. And also, they know that people are going to buy this and spend $60 where they're not going to do it for a twig. But if you plant them like this, this is what they look like. This is not going to produce very much fruit, and the fruit's going to be very, very poor quality. This is a tangle of branches. Why do they grow like this? Because in the northern climates, most of the branches, or a lot of the branches, are destroyed by snow mass, by ice. In the spring, they're the first things you can eat. So the deer, elk, moose, they go along and they eat a lot of the small shoots before there's anything else nourishing that comes out. So the trees that survive are ones that put an enormous amount of growth, vegetative growth, realizing that during the winter they're going to lose a lot of it. And the ones that survive and the ones that come down to us as orchard trees are those that grow vigorously. But you transplant this into a California orchard and it's totally inapplicable. And what we're doing with our pruning is trying to restore some of nature's way of pruning the tree and getting it to be more productive. These trees came from a very nice, actually, garden, community garden. Once they're pruned with several large scaffold limbs, horizontals, and fruiting shoots, they have a chance of producing some fruit. Once you cut, for example, in a peach, once this has grown this past season and produced fruit, it'll never produce fruit again for the entire life of the tree, no matter how long it lives. It's only going to produce fruit on new shoots. It's only good for 1 year. So you prune it back and you have these new shoots and you pick two and let those grow up like a Y. And then after they produce fruit, you cut them down to 1 or 2 buds and let them go again. And then you don't get this huge peach tree where the peaches are so far from the source of nourishment that they become small and shrunken and poor quality. And those of you who go to a self-pick orchard can know that if you come to an orchard now and go back to it 10 years later and pick fruit, the trees are just about the same size. They grow very little. They're kept the size where they produce fruit. So basically you have 1 year's growth, then you cut it down to a few buds and you have the second year's growth. And this all depends on the auxin and controlling the auxin. So this is a very nice community orchard in Jessup. Most of the farmers there are from Zacatecas. They're excellent farmers. Their beds are beautiful, but they plant high density. They have trees all along the garden beds, which is a very good way of growing in the French intensive system, but they don't get any fruit. This is what they look like. So actually, Steve Huffendahl, who's from the Foothill Division of the California Rare Fruit Growers, and I went there and led their farmers in a pruning cycle and got the trees to look like this, with just a few major scaffold branches, nice horizontals, and fruiting spurs. And this is the way they should look. And this is just very poor planning. And it's not a knowledge of what the hormones do and how you control them. Here's an example of this typical strong apical dominance in palm fruit. Pears are very apically dominant. All of the branches need to be spread and weighted. We mentioned why the apically dominant trees grow in this fashion, because their genetics were determined in forests of these trees. So to develop fruiting buds, compact upright trees must be spread to 45 to 60 degrees. When the sunlight hits the upper side of the branches, flower buds are favored. Most of the buds on all of these trees are stem cells. Just like human stem cells, they can develop into any one of a number of different forms, depending on their geography and the light forming on the tree. So for example, if this branch is in shade, these buds will be shoot buds. If it's in a lot of shade, they'll have very long spurs, water sprouts that are an attempt of the tree to get into sunlight. Once light hits these buds, they're more likely to become fruit buds, the same buds, because they're multi-potential. The auxin is gravitationally controlled. So it runs down. When you move this to 60 degrees or 45 degrees, the auxin moves down the underside of this branch and it suppresses all these buds. That's why they don't grow down like that. If they grow down, this isn't in the right position. So it suppresses all the inferior buds, and the upper buds are freed from the auxin suppression. And that's why the auxin is very sensitive to gravity. It layers along the underside of the branches. The upper side buds are freed from auxin suppression and will allow flower and leaf buds to break dormancy. Orchard after orchard after orchard, I find very healthy trees that are just not trained. And it's just, it just doesn't make any sense. And that's why I think it's so important to understand the action of these hormones and how it can help you get decent crops. Using spreaders to train a young pear tree. This is the way pears grow. And yes, it does form some fruit, but nothing like what you would see in a properly maintained orchard. You can use spreaders, you can use weights, anything that gets them down. If you get them down to 45 degrees, they still have some upward growth. Once they have the growth you want them down to 60 degrees or horizontal, or in commercial orchards, they even get them below the horizontal. Here's a standard planting in a 500-acre apple orchard. Every single one of these trees is trained. A tremendous amount of work. But on the other hand, that's how you get 50 tons in an acre. This isn't a high-density orchard, but this is going to be a very productive orchard. And this is what is done. And the reason I emphasize this is all commercial orchards engage in intensive tree training. We tend to be fascinated by using chemicals and fertilizers and watering this and watering that. And the thing that works the best is taking advantage of the hormones and training the trees. For some reason, this is rarely seen in backyard or urban orchards, and the result is surprisingly poor crops in otherwise healthy, beautiful trees. This is properly trained. in high density orchards, all the trees are contract grown and they're treated with a hormone that blocks the auxin. So they develop all these feathers before they're planted. How can we do this organically if we want to grow a compact orchard? And let me show you what we do. This is before it's treated chemically. This is after. This is very dramatic. Use of the plant growth regulator Maxell to induce side branches and create a feathered grafted sapling. The PGR blocks the action of auxin. Well, let me show you what we do. This is a little example before I show you what we do in an actual major orchard. This is called notching or forcing a bud. This is the method that we use to create feathered trees in organic management. It's labor intensive, but that's organic. I'll show you an entire small orchard done with a 1/4 inch rat tail file. So this is a nice little tree in a school garden in Venice. It's an apple tree. It has a nice first whorl, and the second whorl is now coming out, and it alternates with the first one so that one branch doesn't shade the other. This is going to be a very productive tree, but this branch is missing. A tree should fill its allotted space. It's a major objective of orchard management and tree training. Most orchardists in the Central Valley and the big orchards say any light that falls on the orchard floor is like taking gasoline out of your tractor and pouring it on the ground. This is the energy that drives the whole process, and you want it to hit a leaf. The leaf is like a little solar panel. It collects the light energy in the way of photons and actually causes the joining of carbon dioxide and water to form the carbohydrate. And by polymerizing everything else, starches, oils, waxes. So basically, you want the sunlight to hit something. So here's what we did for this little tree. This all takes place in the same season. We used a girdling knife and we cut a notch. You can do this with a jackknife or one of your grafting knives. We cut through the bark, through the cambium, and through the phloem, the green, down to the xylem. So the xylem is like the tree's arterial system. It's carrying all the nutrients from the roots up into the leaves. The phloem is carrying the products of photosynthesis, the tree's metabolism, down to the rest of the tree. And here is a dormant bud. And it's dormant not because of the season, it's the early summer. It's dormant because it's in paradormancy. Now, this is about a week or two later, this bud comes out and it forms a beautiful sprout and a branch. This is 5 weeks, the same season. Now, here's the key. Here's where the notch was. The notch is already healed. I painted it so I could, because I go to so many orchards, so I can come back and see what happened. Here's the thing I want you to see. These are the rest of the buds. They're the same age as this bud, but these buds remain dormant. They're in paradormancy, and they're in paradormancy because the hormone auxin is still flowing down this branch and blocking these buds from blossoming. But this one, as soon as the auxin is removed, it comes out and forms a huge branch. Here it is being spread, and that tree will be in its final shape by the end of the first season. So this is dramatic evidence of how the hormone works. And if you just remember this little example, you've got a tremendous clue of how you can use this information. This is that branch in the same season. If you have an espalier, it's missing a branch, and you have some healthy buds that can be activated, you can use it in many, many instances. All right, here's a good example of what we did in an orchard. This is in Pasadena. This is the back of Huntington Memorial Hospital. This is an 1.5 acres, a very, very nice garden. They wanted trees, fruit trees, but they didn't want to shade their garden beds. So they said, what do you mean? We don't have room for an apple tree in our little garden. This is a row of compact, a high density line of 25 plus apple trees. Takes up less than 3 feet of space along a sidewalk fence at the Waverly School Farm. It was planted, trained, and pruned by the orchard team with the students and the teachers and the families. Now, these were bench grafts. That's the size of a number 2 yellow pencil with the eraser is a single scion that's planted in a cleft graft, just like you guys do in January and February. This thing was just the size of a pencil. In 1 year, it grew to the top of that fence, but it had very few side branches. A few of them did. They were just straight. they're very strongly apically dominant, and they wanted to get up into the light because there is shade from these structures, the hospital, these palm trees. So in February of the following season, each kid had a 1/4-inch rat tail file and notched. Remember, the buds go up every one of these trees in a spiral fashion. They point in every single direction. So they picked the ones that were pointing this way and that way, north and south, made a notch right through down to the white through the phloem and cambium, right down to the xylem, every other bud. And this every tree became feathered by late February, early April. So what happened? The school wanted to have an annual cider festival. The second season, and actually Jeff was here to plan to prune these, which were growing, unfortunately, not on dwarfing rootstock. We had a real pruning fest. This fall, they had a great harvest, a fresh eating apple for each student. They filled 50-gallon jugs with fresh squeezed cider. They filled 24-quart bottles of apple pie filling and had 4 more baskets of apples for the parents and visitors. So this is what it means to adopt commercial techniques to small orchards, not using the hormone, which would have been a cinch, but doing it organically with just rat tail files. All right, when you get these branches horizontal, you get tremendous production because every one of these is hit by the sunlight. They blossom out. The fruit is beautiful. And it just depends on using this information. There's a large crop. No branch is allowed to get more than 50% the diameter of the trunk. That's it. Then they're cut using what's called a renewal cut. It's called a trapdoor cut or some other inelegant terms. But nevertheless, this frees up the epicormic buds, and within the end of one season, you have another producing limb with buds that are in the sunlight, and by the following year, you have your fruit. Very dramatic evidence of what this information does for you in your orchard management. All right, now what about grafting? How does auxin control influence 4-season grafting? Although in the California rare fruit growers, we primarily emphasize forms of cleft grafting, T-budding is the world standard. And why? Because T-budding can be done all through the year because the buds are in paradormancy. You don't have to wait for the dormant season. But the dormant season is easy, and in February and March, you have in January and February, you have your scion exchange and you do your cleft grafts. Here's a bud graft. So you make your T down to xylem, you lift up your flaps, and you put in, here's the dormant bud in paradormancy with its petiole. All right, so what happens? When we do June budding in the coastal part of the county, 7 days later, we transect the stem 3.5 inches above the bud. If there's no leaves above and below the bud, the stem is crippled. And what that means is we just bend it over so it's horizontal or below our bud graft, because this branch that's above still has leaves, and they're photosynthesizing and producing carbohydrate to increase the dynamic growth of that bud. You don't want to take that away, but you don't want it to be suppressed anymore, so it can be crippled or bent over. 10 to 14 days later, the stem is transected 1/2 inch above the bud. We're now out 3 weeks from the budding date. We remove the budding rubbers. When the new shoot reaches 10 inches, we remove all the shoots and suckers. So this technique allows the auxin to protect the bud from leafing out before an adequate callus and preliminary vasculature is developed. One thing you see, even in the rare fruit growers, when the people who aren't experienced, they get a beautiful scion, they put it in their root, in a rootstock, and it buds out beautifully. But within 2 weeks, it's gone. It shrivels and dies because it's budded out too quickly. As soon as you remove that auxin suppression, that bud is ready to go and has enough energy to bud out. This is after several weeks, but it takes time for the callus to heal and for the phloem and xylem connections to develop. So the auxin protects the bud from leafing out before an adequate callus and preliminary vasculature is developed. It also permits continued photosynthesis in the remainder of the specimen and then encourages growth of the sprout. Very important, critical thing in bud grafting. All right, let's look at the gibberellins. They promote cell elongation, cell enlargement, and cell division. They keep track of the photoperiod and inaugurate flower development and influence bolting. They're growth retardant and acts in many instances. Growth retardants act in many instances by blocking gibberellin. Very often, trees that are planted under power lines, when they reach a certain size, are sprayed with gibberellin that leaves the tree entirely intact, but discourages it from further upward growth. So gibberellins can maintain dwarfism, which can be reversed by using a gibberellin antagonist. Gibberellins facilitate fruit set and enlargement as well as parthenocarpic development. Here's a key. Some gibberellins released by the developing fruit seeds suppress downstream flower development and fruit size. They promote alternate bearing. This is a very important mechanism, and some trees have it more than others. I mean, if any of you have grown some of the newer mandarins, the golden nugget, for example, you can have baskets of fruit one season and enough to fill a fruit bowl the next season. They're very, very much influenced by gibberellins. Gibberellins help the tree have a rest year between big crops because trees tend to flower out dramatically in the spring because they've got to attract the insects. They've got to attract bees and syrphid flies and other insects that are going to pollinate with all the competing trees around them. So they produce many more flowers than they need. And that's why they have fruit drop. In July or May in the coastal regions, the tree will drop a certain amount of fruit depending on what's called the carbon ratio in the tree. The tree has a way of calibrating how much resources it has to bring those fruit to maturity and the seeds to maturity, and will drop the remainder. Your citrus trees routinely drop about 90% of the blossoms normally because the tree produces many, many more to ensure that it's going to get some fertility and some pollination. And that's controlled by the gibberellins. As soon as the seed forms, the gibberellins begin to be developed and they go down the branch and they suppress flower development. And it varies from tree to tree. This slide, it explains why you have to have a certification in pesticide registration to use them, because they're very potent, these hormones, and when they're sprayed, they can interact with other orchards, and that's why they need to be controlled. But we can control them ourselves, and we can control the adverse effects of gibberellin in our small orchards and reduce alternate bearing. Here is a peach tree, excellent fruit set throughout the tree, thousands of fruit. Thinning, a most important orchard practice, often neglected, leading to small fruit and alternate bearing. The tree's objective is to produce a seed, not to produce fruit. I mean, the seed-fruit ratio is going to change dramatically. It'll only produce enough fruit to get that seed carried away. That's why it produces a coating around the seed. The seed is its responsibility. The coating or the fruit is what it uses to get some animal to carry that away and not compete with the mother tree. Thinning avoids alternate bearing. This is much too late for this, but it's a good example. In commercial orchards, it's done right after the blossoms are fertilized, but most of us are not skilled enough to determine that, and we're not using hormonal thinning. When it reaches pea size, that's when the gibberellin begins to be formed, and that's the best time to thin. This is the first pass. This tree got 3 passes to get down to that. And the result of this is from a whole bunch of small peaches to an excellent crop. This is in school gardens. This is May Pride, the wonderful peach harvest in May when the kids are still in school. This is earlier than almost any commercial orchard can produce in early May. This is by the 12th of May. Apples, they're thin to 1 or 2 per cluster, the same thing. And many gardens, especially where people are farmers and they say, well, we're losing a lot of fruit, that's a lot of fruit. Well, it isn't. When you weigh the difference between the fruit and the seeds are what the tree is going to be sure reach maturity and size not the fruit around the edges. All right, enough of that. This is the last slide of hormone. I'll tell you a little story that's interesting. Ethylene is a highly volatile, relatively unreactive gas produced by all plants. Ethylene is involved in the transition of fruit from being physiologically mature to being ripe. Physiologic maturity is when the tree decides genetically that seed is embryologically mature and can produce a shoot. Ripeness is a separate category when more sugars are developed and the outer fruit coat expands and softens. The ethylene also prepares the fruit for abscission, that's falling off, and enhances the final coloration. Ethafon is a liquid commercial product that releases ethylene. It is routinely used to ripen bananas and avocados. Those of you know, avocados never ripen on the tree. Or almost, you shouldn't say never say always, but it's very rare that avocados will ripen on the tree. The longer you leave them, the more oil they accumulate, and then they become rubbery because they have an ethylene blocker in the tree, and the avocados produce it very, very slowly. They have to be removed from the tree and stored for a while. And that's why the commercial avocados are in cooling sheds, and when the market is ripe, they dump in Ethafon, and it ripens within hours or days. How it's used in orchards: one application in a walnut orchard enables the farmer to remove the walnuts with a single shaking, because what it does, it prepares the fruit for abscission. So that's called a once over harvest. That's how it's used commercially. Folks like us and small orchard growers use very well-known techniques to stimulate ethylene and ripen our homegrown or store-produced fruit. Retain is a commercial product which is used to block ethylene and prevent premature fruit drop. It's used, among other actions, to control the harvest date. Why do we put unripe climacteric fruit in a paper bag with a slice of apple? Apple has a huge amount of ethylene. Let me just show you the ethylene and the ripening of climacteric and non-climacteric fruit. Those that can ripen after being picked are fruit that produce ethylene on their own in enough quantity to go from maturity to ripeness. Those that can ripen after removal from the tree are non-climacteric. So if you watch a climacteric fruit over a period of time, the graph of ethylene corresponds with the development of the actual fruiting layer, color, aroma, sugars, and flavors. And the color break stage also has a little bump in the ethylene production. This is a typical chart from ANR, Agricultural Natural Results. It shows the climacteric fruits and non-climacteric fruits. And there's a tremendous range. The cherry moya and the mame bananas all require ethylene to ripen. It's really interesting. Ethylene was used back in biblical days. It was used in ancient Egypt. There are carvings on walls of buildings in the Pharaoh's day, showing people ripening grapes and figs with smoke from sheaths of grain. It was used over 1,000 years ago in India to ripen their fruit. When they had the fires in Ventura last year, that farmers noted that the cherry moyas were dropping off the tree about 2 weeks before the natural harvest date. Fruit was ripening prematurely in the fields, and fruit was dropping from the trees because ethylene is a very significant component of smoke. And in fact, the earliest characterization of the chemical ethylene was in Russia and in England when they had gas lights. People noticed that when there was a leak in the gas tubes that went up to their lights in the building, the plants that were on the windowsills there would mature very rapidly, become senescent, and die, only where there was a leak. And finally, a graduate student actually characterized using gas chromatography that ethylene was the component in the actual lighting gases. All right, in concluding, I wondered if I successfully made the point. Research and new understanding of the plant hormones enables spectacular results of precision orchard culture. Can we use the same knowledge, replicate this on a human scale with a sense of accomplishment and satisfaction in the care of our backyard orchards? Well, a quote by Yves Chouinard, who's been in the news recently because he donated his company. He was the founder of a very famous clothing and outdoor company. The more you know, the less you need. So to increase fruit size and orchard production, ProGib LV Plus is applied by helicopter and avocado orchards. Can we do the same with a simple tool? I recalled a couple of slides on a little backyard orchard story that might just illustrate the simple message. Will this increased understanding of the plant hormones better inform our horticultural techniques in our home orchard? You be the judge. So here's this little story, my last two slides. Called to see a charming lady with a lovely holiday avocado tree in her backyard orchard. Best avocados you've ever tasted, her husband said, but not a single avocado in 7 years. Please come and see it before we cut it down. There's a tool. So I called one of our very, very famous farm advisors, Ben Faber. And this reminds me of a quick story, since I got a few little minutes left with Julie Fring in the audience. It's kind of like, when one of the famous survivors of the Johnstown flood in Pennsylvania, when he died and went to heaven, he went to the desk there, and St. Peter said, welcome. We know you've had a very difficult life in a flooded town and all, but you're welcome. He says, well, St. Peter, I've been telling the story about the Johnstown flood over and over and over again for the last years of my life. I'd rather just tell the story once more and get it over with. And so St. Peter looked at me and says, well, yes, we do. He says, do you have an auditorium? He says, yes, we have a large auditorium. We can invite everybody. They can come and hear your story. But I got to remind you, Noah will be in the audience. Anyway, I just thought of that when I saw Julie Fring's portrait go up in the gallery there. So here I talked to Ben Faber, and I said, what should I do? I went and I saw the tree. It's healthy. It did a leaf analysis. It's got good nutrition. They water it. They pruned it properly. He says, well, it happens. They do. Why don't you try and girdle one of the major branches and concentrate the hormones, particularly the gibberellin, in that branch? So I took a girdling knife. The girdling knife has two blades and a little U-shaped blade at the end. And the knife is right there. And you can draw that around the tree, and it'll cut a beautiful, even 1/4 inch or 1/8 inch, depending on the size of the girdling knife, groove right down to the xylem. So there it is. There's the notch. That's the completed girdle. All right. No word from the lady for over a year. Ben was scheduled to talk at Huntington, and I was invited to go as well. I knew he'd ask me, how are things going? Did anything happen? So I thought I better get in contact with the lady. So I sent her an email, and she sent me back a 2-word email reply. It says, we're pregnant. So I went and saw her. You can see it's a small tree. It's what, 6-7 feet tall. She's 5 feet tall. The girdle is well healed. They heal very rapidly. And then you get a flood of hormone down that branch. So she had these two, this is a holiday avocado, obviously. It had these 2 big avocados. I was a little disappointed. I said, well, it's better than nothing. And she said, well, no, these are for you. I said, no, you keep them. You've been waiting 8 years for some avocados. I have my own avocado trees. She says, no, you get it. Come with me in the kitchen. I'll get you a paper bag. So meanwhile, back in the kitchen, she has her crop on a little kitchen table. So that's what I was confronted with. Pretty dramatic, I thought. Well, that brings us to the conclusion. I hope we'll have time for some questions. I certainly appreciate your attention, and I hope you found the hour worthwhile. Many thanks for the invitation. All right, I'm going to stop sharing now. And Jeff, I'll open if I can do it, I'll,
Okay, you just want to stop sharing? There we go.
okay. Am I okay? Am
Yeah,
I here?
yeah, you're back. Yeah, we're back to
Okay.
to seeing you.
All right. I can try to answer some questions for you. I know it's evening time.
Okay, John McNeil, are you going to handle the questions or just going to
Yeah,
have people
okay,
unmute?
Khalid, if you're there, you might want to introduce your question directly, because it's a big one.
Yeah, it's a long one.
Oh, hi, that was a fascinating talk. I really appreciated it, and it really got me thinking about all the things I'm doing wrong and some of the things that turn out right seem to happen by accident. I guess it's by design. My question is I've propagated hundreds of plants, and I've noticed, especially with figs, that sometimes for the same variety of fig, for the same scion from the same source, I typically divide my wood into multiple pieces. When I root or graft, but let's say we talk about roots, so that each scion has its own set of roots. Some of the scions grow better than others, irrespective of cultivar. The ones for me that seem to grow the best, that show the best vegetative growth in the first year, are the ones that seem to have larger or longer internode spacing. The ones where, when I look at it on the tree, I would say it's not going to produce fruit, or it's not going to produce much fruit. The scions that don't seem to grow that well are the ones that seem to have very tight node clusters, the kinds that I would see in a mature tree at the very tip of branches that I would say are perfect for fruiting, that might produce 12 or 15 fruit per cluster, but may not grow at all during the season. So I guess my question is this: have you noticed that scions that we use for propagating carry with them their hormone profile and can influence or be influenced by that profile in the way they grow post-graft? And if so, what should be growth?
Lost you, Khalid.
Yeah, Khaled, we lost the last 30 seconds.
I got you. Yes, it's very, yeah. The interestingly enough, yeah, of course, the scion, each cell of the scion has all the information to grow the entire tree. Every single part of the tree is already in every single cell. They don't have separate germ cells, so that the somatic cells have all the information to grow. And it really makes a big difference. The difference in the rate of growth of the scions really depends on the carbohydrate storage of that particular piece of the tree. So basically, those that have had a lot of light and have been able to build up carbon stores, they're the ones that are going to root out fastest. The internode is, of course, the suppression by gibberellin. Actually, it can maintain dwarfism in the tree, and the stage of when you remove that scion is very critical. One thing I notice, I go around to a lot of orchards and community orchards, and a lot of these people who have migrated from Central America, they grow trees that they're familiar with. They grow avocado trees very frequently. Almost every one of the low-income housing have gardens with them. They're beautiful trees, 5-10 feet tall, maybe longer, been growing for 5-10 years. They never get any fruit. The point is they're planted from seed, and until the only reason that you should grow that from a seed is to get the rootstock. Then you cut it off, and then you take a scion from a mature tree that's already gone through adolescence. An avocado takes 12 to 14 years before it reaches the point where it's going to set fruit. An apple, 4 years. Every tree has that built in. And various varieties have a different latency period before they're old enough to actually set fruit. And so the point is that you're taking a mature scion and you're putting it on vigorous new rootstock, and you're getting a tree that's characteristic of the scion, not the rootstock. So basically, you want to choose scions that have had the benefit of good sunlight and of good storage of carbohydrate, and that are low down in the tree. And they have to be new shoots. They should be ones that are that season or the season before that. That's when those buds are most likely to break dormancy and form roots. Those that are over a year old, the buds are already drying up, and they're usually only good for 1 year. There are epicormic buds underneath that you don't see, and it's not the bud that's on the surface, that dried little bud that's leafing out or rooting out. It's the epicormic bud underneath it. But nevertheless, it's a good indication of where there may be epicormic buds. But basically, it's the amount of light that shoot has been exposed to, the age of the shoot, and the position on the tree that's going to govern its rooting. If you use rooting hormone, it's apt to even out the rooting rapidity of the different scions, but they are going to reflect their origin. And I don't know any other way you can actually tell exactly when it's going to bud out. I mean, it should leaf out fairly promptly once you get it in the right rooting environment. But that's the best I can tell you about that. It's the amount of carbohydrate stores in that. If it's still somewhat supple and doesn't snap and break, it's probably good for propagating. If you're propagating hardwood propagation, and also the distance between the internodes, I'm not sure whether that tells you very much about what type of tree you're going to get. It's usually the age and the season that you collected that scion wood, whether it's been stored or whether you're using it fresh. If it's not stored properly, it's going to use up its carbohydrate stores. It's going to be metabolizing as long as it's still alive. So it does depend on being stored at the proper temperature. Some of the scions need to be what's the term for, you need to buy a season by cooling them for several months, but stratified. They need to be stratified. But most of the trees that you're propagating don't need that. They should bud right out after you get them fresh. Anybody else can chime in. That's my experience, is that the shoots that are low down on the tree but have been exposed to sunlight and have a good carbohydrate store by their size, and they're not very old, are the ones that are going to root best.
Yeah, that's essentially kind of what I'd seen, but I haven't been able to. That's just my empiric observation. Was even if the tree, a lot of times the trees, when they start to fruit, they just stop growing. At least the figs do. But if I notch them and I can get a bud from lower down to bud out, they grow very quickly. But they'll fruit maybe very sporadically, at least that season. So I was more interested in propagating. So I've noticed that in propagating, the wood that to me looks like it's going to grow quickly and not necessarily fruit seems to be the ones that give me the most growth the first year. It's almost like they maintain that profile for at least another year. So that it's helpful to hear a more scientific, rational explanation of it.
Yeah, once it starts fruiting, the hormones are preparing to shut that tree down. So you've got to get at it earlier in the season of its growth. When it's first budding out and reaching maturity before it sets flowers and fruit and uses up some of its carbohydrate stores, in the earlier period, you're more apt to get rapid rooting.
Okay, I think that's answered that question pretty well. Cindy's got a quick question, and I think it refers to girdling. With the technique that you used on avocados, the girdling work on other trees like mangoes, for example?
I've never done it on mangoes, but girdling is a very common procedure that's used in Europe very, very frequently. Even when I was a young traveling student, I would visit orchards in France, and they would try to girdle a branch on each tree to develop fruit for a special market or for a fair. You want to only girdle one limb. Once the girdle heals, it can be girdled again, but you move around the tree because you're denying the rootstock and the storage capacity of that tree by girdling it. You're backing up the stores in that branch. So you have to be careful not to girdle a major trunk or 2 or 3 limbs that are supplying that tree with nutrient. And the time of year is critical, but it's used most often to produce larger fruit throughout Europe. I haven't seen it used very much in nurseries here. I'm sure farmers who bring their produce to a fair or to some sort of market competition are very knowledgeable about every different variety. But most fruit will respond to girdling, especially early in the development of the fruit. And using it to jumpstart a tree that's gone into this semi-dormant period. Apparently, according to Don, Hodel, they very often will do that for no reason whatsoever. And it may not be a group of trees, but it may be one in a group of trees that all of a sudden stops producing. So this type of girdling does tend to give a flood of hormone that actually can get most of the tree into a producing stage, not only the limb you've girdled, although if you girdle a tree for increasing fruit size, when the fruit is starting to form, it'll only affect that single limb. And basically, it has to be done early in the formation of the fruit, and it can lead to dramatic improvement in the fruit size. So that's when it's most commonly used, and it works in most fruit. I don't know that it does in all of the tropical fruits, but it certainly does in all of the temperate zone fruits.
Okay, so is it done before the flowering occurs or after the flowering occurs? Well,
it should be done after you see how limbs have been pollinated. You don't want to girdle one where there's been very few flowers that have been pollinated. So once the fruit begins to form, there are various times with various varieties that are best. And you have to experiment or talk to somebody who's used to girdling that particular variety. But generally, once the fruit begins to form is the time when you want to build up the hormonal stores and the carbohydrate stores in that limb. Before the blossom forms or during blossom time, it's hard. It's hit and miss because they're very often pollinated erratically, unless you're in an orchard where you're using your own beehives and you know that you're getting pollination of most of the flowers.
Okay. Yeah, I think that probably answers Irene's question that she's got here. She's got an avocado that's got 2 grafted branches, a Forte and Mexicola. And she's wondering whether she should girdle both branches to induce fruit or just one at a time.
Are they trees that have stopped producing?
She doesn't say.
Yeah. Well, if a tree has gone into this rest period over several years and you can't find any other explanation for it in its fertility or in the way it's being pollinated, then girdling is kind of a last resort. But it's best to be done on one major limb, and because it'll build up the carbohydrate stores in that limb dramatically, and also the hormones that are produced will be concentrated in that limb. And generally, when the rest of the tree is ready to bud out, that tree will have the necessary stores to do it. Once the girdle heals, there is a flood of that enzyme, those enzymes and nutrients into the rest of the tree. And it very often will affect, in this particular instance, in this pseudo dormancy period, it generally will affect more than the branch that you've girdled. But it's erratic. It's something to keep in your back pocket. It's an important technique. It's one of the things, with biological systems, there's a lot of variability and there's a lot of issues that go into each tree. But I think in this introduction to the hormones, you at least have some scaffolding to put more information and observe what happens when you do various things and try and link it to what the genetics of the tree is requiring and what the hormones enable you to do. So this you build on this information, but a lot of it is relatively new. That most of the information that has handed down to us is observational. A lot of these things have been observed for millennia without an explanation, but people know about them and they use the techniques. Like it was only in the 1800s, early 1800s, that ethylene was even categorized, even though people knew that there was something in vegetative smoke, in forest smoke and in smoke of straw that will stimulate senescence in plants and maturity and ripeness in trees, without understanding what was in the smoke. So these things are being categorized, and the newest thing that's available, really only in the past few months, is a packet that's about the size of a tea bag that I think will be available commercially in retail nurseries as well, that you can drop in to a crate of fruit that you want to keep from ripening, and it will keep that fruit from ripening for about 2 or 3 weeks. So it's been used now rather than blocking ethylene, because it's a lot easier to use. It's not a gas. It doesn't require special storage equipment. I have that on my last slide, what it's called, but it's very new. The licensing or the restrictions on the PGRs for blocking ethylene have expired, and so many other companies are getting and making these dry products that can be used very easily. So more information will come out about this as time goes on, because many more companies are getting involved, and as they develop these plant growth regulators, it tells us more about how these normal hormones work in the trees that we're taking care of.
Okay. We got a few more complicated questions. Can you manipulate alternate bearing by grafting from 2 different sources that are alternately out of sync as far as their bearing is concerned? Will they synchronize in time?
Yeah, that's a very good question. That's probably most professional orchards, most commercial orchards use that technique to change the synchronicity of the tree. And for example, in mandarins in the Golden Nugget, by pruning aggressively different parts of the tree, you get new shoots that will bear alternatively, and they will change the synchronicity of the alternate bearing. So you could do it by grafting, but that's a longer process. You can do it by aggressively pruning the tree, and pruning it in various ways by doing thinning cuts, not heading it, but by doing thinning cuts and stimulating new branches. It's not the true thinning cut where you're cutting it at the bark branch color, but you're doing a renewal cut where you leave about a 2 to 3 centimeter stub that's angled like a trap door. And within a season, you'll have a new branch that is at a different synchronicity than the rest of the tree. So that's the general way one breaks this alternate bearing if you happen to get into it. The other thing with the temperate, not the semi-tropical trees, but with the temperate zone trees, it's doing early thinning. That will absolutely break the alternate bearing, because the gibberellin isn't produced and does not suppress these new flower buds. So those are several techniques. Grafting a new limb would do it, but you're waiting several seasons. But a very good question, and it's very important to think through these things and decide how you're going to do it. But obviously, we have a lot more control over the orchards the more we know, as Chuinan said. The more we understand these things, the more techniques we can bring to bear on them.
Okay, well, that's absolutely fantastic information. I'm just wondering, we're lucky enough to have your presentation, but is there any publications that you might recommend that concisely say what you've told us that we can use as a reference later on?
Yes, I think it's interesting. There's very little talks you can access that treat the hormones as a whole, like this, like I put together today, that kind of puts them in one framework. There's a lot about each individual hormone, depending on whether the article is about harvesting or if it's about grafting. But ANR, the Agricultural Natural Resources, has a brochure that they use for training people before they get their pesticide certification, specifically for use of the plant growth hormones. And it's a plant growth hormone manual. And I found that to be an excellent reference. It's a small manual, and it's probably available in the ANR catalog. I don't have the thing readily handy, but it's a paperback preparatory course outline that goes through the use of the PGRs, the plant growth regulators, in all different crops: row crops, vegetable crops, fruit crops, landscape trees. I think that's a very good place to start. The idea of showing these examples that I showed you, I haven't found anywhere. And that's why I decided to put it together, like demonstrating notching and demonstrating the use of using the notching to produce feathered trees. You won't find it in most of the organic manuals, although it's a great organic technique. But I think that's the place to start. It has a lot of references, and it gives you, like I've been trying to do with this, enough background material that you can reach out to other articles and find it. But there's much more on the PGRs than what I've tried to discuss today, using your own intuitive and your own initiative to use these hormones in orchard management. But I think it's a growing subject.
Yeah, well, thank you very much, and we're lucky to have had you give the presentation tonight.
I think I found I put a link into what I think is you were talking about, plant growth regulators on the UCANR catalog site.
Yes, there is one. It's in a site that was used during the early days of the COVID virus. It was called gardening. It was a whole series of videos, Gardening in Place, I think it's called, by the Los Angeles County Master Gardener Program. And it has a whole bunch of very eclectic lectures about things that can be done in the home garden alone. And that lecture that you recorded last time in its entirety does appear on that. And so that's one. It's already up and running. And this one has a lot of slides at the end that discuss exactly how photosynthesis takes place, and a lot of things we use in our lecture that I use in the orchard courses for the Master Gardener program that I've given in Riverside and Los Angeles, that actually shows how, I was stimulated by the fact that people were saying, well, it's the orchards that cause all of our droughts, and it's growing all these almonds. And I said, an almond tree doesn't use more than 2 quarts of water for an entire season, and it's not watered 6 months out of the year. All that water is picked up and it's transpired, it's purified by the tree, and it goes back into the atmosphere. The problem is pipelines. It's bringing the water from where it falls to where it's needed, but it's never lost. And you know, you have to really see where the issue is, that trees have to have that water. It's one of the defects in trees, is that they need to keep those stoma open in order to get air into the leaf to form carbohydrate. And it's just like a solar cell. The photon hits the chromophore with the photosensitive pigment, like chlorophyll, and the electron is activated. And rather than going around like a solar panel and operating a battery and engine, it goes into a higher orbit. And when a molecule of water and a molecule of carbon dioxide come together in that leaf, it sparks and joins the two together and goes back. And it's never lost. But that stoma has to be open. And when it's open, the tree is losing its transpiring water that's purified back into the atmosphere. But if you take all the things, the leaves, the nuts, and you weigh them at the end of the year, that tree has accumulated a quart and a half of water. That's it. So there's a very good slide that shows this whole process that I try to educate our 50 or so yearly master gardeners. But anyway, so there are a number of slides at the end that relate to different subjects that I keep in reserve. But that website, it's called Garden in Place, Los Angeles County Master Gardeners. And there is that video. It's about 2 hours long on plant hormones.
Okay, well, thank you very much. Appreciate it. Jeff, I'll hand back to you for now, and you can wrap up and then introduce me again so that I can give my chairman's award.
Okay. Well, Herb, thank you very much. I did put a link to the Master Gardener YouTube channel in the chat, and the Garden in Place talks are one of the playlists you'll see there. And there's an hour-long talk in there that you did, Herb. I don't know if the one you're talking about that's 2 hours is there. Some of the stuff you've recorded for the Master Gardeners is not available to the public. So, but all the Garden in Place stuff is. So hopefully that's the one you're talking about.
Yeah, I think the hormone talk is in there because it's
okay.
Yeah.
Okay. Yeah. Actually, today's talk is actually on that site too, in a different playlist. But you know, your earlier version of it. And I know you cover some different things this time, so heard some new stuff myself. So I really appreciate that. Well, thank you very much.
Thank you. I'll let you give your awards now, and thank you very much, ladies and gentlemen.
Okay, John McNeil, you're back.
Okay, thank you to Herb. I appreciate it. I'm trying to center this, but this is a plaque that I am going to award to a member of our chapter that has done outstanding work. You can see the workmanship that's gone into this plaque. It is outstanding. It's a real piece of art. Anyway, during my chairmanship, I was granted permission to create a chairman's award to be presented by the outgoing chairman to an individual who has made a significant contribution or has made significant contributions to the chapter and is approved by the executive committee. Tonight, I am pleased to make the first presentation of this award. A little bit of preamble. Before being convinced to join CRFG by Eunice Messner, our honoree was a member of the Orange County Organic Gardening Club and actively participated in that organization by writing many articles, in excess of 100, for their club newsletter. In 2002, this person joined the OC chapter of the CRFG. With leadership qualities that soon became evident, this individual served as a chapter chair from 2007 to 2009. And of course, in 2008, we were the host, a very successful host of the Festival of Fruit, and that was the year of the avocado. Shortly thereafter, mentored by Denny Luby, another prominent contributor to the chapter, our honoree assumed the position of Green Scene Chair, and with Denny's continuing and lending all assistance possible, continued and even improved the practices that had contributed to the Green Scene's significant success. As you know, thanks to its Green Scene sales, the chapter has been able to award generous scholarships to students at Cal Poly Pomona and Fullerton College for more than 2 decades. A wonderful achievement. During these years, our honoree has chaired the Green Scene sales, and it's estimated that, we've probably generated $100,000 over those years. In 2015, his prolific articles sharing the wonders of his impressive garden, often written tongue-in-cheek, so to speak, attitude, and always accompanied by beautiful photographs, have educated and entertained thousands of Fruit Gardener magazine readers. Our awardee is a fountain of knowledge, ever willing to share his experience with anybody who has questions. He never hesitates to volunteer for booth duty at the OC Fair, working a minimum of 5 shifts every year and often more. He contributed thousands of plants over the years to help out the annual sales success. And for many years, he was a featured grafter at the chapter's annual grafting demonstration meeting. He has made available his extensive garden for training purposes such as pruning, wood collection, and grafting. He serves as an expert advisor to the chapter's propagation committee, and he generously shares the seasonal fruits of his garden for members to enjoy at the tasting table. Please, by now, of course, there's no confusion as to who the award is going to go to tonight. So I'd like everybody to give a hearty round of applause to Don Winterstein. He's my selection. Don, it's very, very difficult to come up and collect this award as was originally planned, but I definitely will get it to you over the next couple of weeks or whatever. But the award, let me take the tape off so that everybody can see. But it is quite a significant award, and you can see there the Chairman's Award of Honorary Life Membership to the OC chapter is presented to Don Winterstein in recognition for his outstanding contributions to the Orange County chapter and to the California rare fruit growers and to CRFG Inc. On this day, 15th of September, 2022, by the past chairman, me. Okay, so and it's a significant and a beautiful piece of art. I tell you, full credit goes to Jay Cruz of the Organic Gardeners Club. He has done outstanding work, and thank you to Martha for the recommendation to utilize Jay's abilities. Absolutely outstanding. Thank you very much. So that's about it, folks. That was I don't think it's the inaugural award. I think in the past, many of these awards have been given to prominent members, but that was way back in the early days. This is now post-pandemic, and as I said, I think it's good. There are so many other outstanding members in our chapter that do make unbelievable contributions year in, year out, every year. And I think it's time that we did something as an executive board, as chairman, past chairman, to recognize these people that have made contributions to the chapter. So I thank you for the opportunity to be able to do this. And I'm glad, Don, that I hope you'll accept this the way it was intended. And if you'd like to say something, that's open to you.
Well, the only correction I would make is that I was the chairman 2008 and 2009, not 2007.
Oh, okay. Okay, but you were there when the Festival of Fruit was on.
Yes, it was started under the preceding president, though. But the Festival of Fruit actually occurred when I was just starting out as president, as chairman.
Okay. Well, you had to see it to fruition, so accolades for that, because it's no mean feat to be able to put all these things together. And you know, as we're well aware, the contributions to the scholarship program has been well received by lots of people. So thank you very much for your contributions, and we look forward to many more years of continued contribution.
Well, thanks for your good opinion.
All right then, Jeff, over to you.
Okay, well, that wraps it up. I'll just mention that our meeting next month is going to be on jujubes or jujubes, depending how you like to pronounce it. We have a speaker coming to us from New Mexico State University. I don't have her name right in front of me. I know I'm pronouncing it wrong anyway, so I'll find out how to pronounce it from her and be able to do that next time. And we'll be most likely it's going to be a virtual only meeting since our speaker will be coming from New Mexico. That's it.
Okay.
Good night, everybody.
Yeah. Thank you. Take care.